Okay, so get this right. You know how your DNA is like this giant library?
Right.
Well, some sections are totally blocked off, like sealed up with ancient genetic caution tape.
Huh.
And guess what might be hiding in there?
What?
Remnants of ancient viruses? Talk about skeletons in the closet.
Yeah.
These are more like ghosts in your genes.
It really makes you think about how our cells have become experts at managing this massive genetic library, you know?
Yeah, for sure.
I mean, we're talking about gene expression here.
Right.
The incredibly precise system that decides which genes get turned on to make stuff and which ones stay silent.
It's not enough to just have the blueprints, right?
No,
Something has to decide which pages to read and when. Like imagine trying to build a house if every single instruction manual from every era was thrown at you at once.
Exactly. And that's why this new research we're diving into today is so mind-blowing. It really sheds light on how our cells keep those ancient viral sequences in check, and trust me, it's fascinating.
Okay, so let's unpack this. Our trusty guide, Science Daily, points to epigenetics, which I know sounds kind of intimidating.
Right.
But think of it this way. If your DNA is the hardware, the actual physical stuff, then epigenetics is the software. It tells that hardware what to do and when.
And like any good software, there's code, right?
Uh-huh.
In this case, it's all about these proteins called histones. Imagine them as those little spools that keep your headphone cords from turning into a tangled mess.
Yeah.
Histones do the same for your DNA, keeping it organized and compact.
Okay, so far so good. So we've got our DNA library, our software deciding which books to read, and now these little histone organizers keeping everything tidy. But how does a cell actually write instructions on these histones?
Well, that's where things get really elegant. You see, histones have these little tails sticking out.
Okay.
And these tails have these modifiable sites. Think of them like sticky notes that the cell can attach or remove, signaling whether a particular section of DNA should be read or ignored.
So we're talking about sticky notes on our DNA.
Essentially.
That's both brilliant and strangely hilarious. But what makes these sticky notes so important?
Well, this new research zoomed in on a specific histone called H3.3
Okay.
and two mysterious modification sites on its tail, K9 and K27.
K9 and K27. Got it.
Scientists have been debating whether these sites carry specific instructions for gene expression or if they're just random scribbles. Turns out it's way more exciting than that.
Okay, I'm on the edge of my seat here. How did they figure it out? Did they, like, invent a microscopic sticky note reader?
Not quite. They used a really clever trick. They mutated those K9 and K27 sites in mouse stem cells. Basically, they slapped a do not highlight rule on those specific sections of DNA, preventing any modifications from happening there.
And then what?
Things got very interesting and very quickly. The stem cells started acting really strange. They had trouble differentiating, like deciding what kind of cell they wanted to grow up to be.
Right.
Their growth got all wonky, and some of them even started dying off.
Wow.
It was clear that messing with K9 and K27 was a very big deal.
Okay, so playing around with these sticky notes on our DNA is not just some cosmetic change. It has some real consequences.
Absolutely.
This is where it gets really fascinating, though. The study found that by messing with these sites, genes that were supposed to be silent suddenly switched on, including some usually found in the immune system.
Right, and that's a huge clue. It suggests that K9 and K27 aren't just random. They play a vital role in maintaining the very identity of a stem cell. It's like they're the guardians, making sure those stem cells stay on the right path and don't go rogue.
So we've got these histones acting like librarians, mysterious sticky note codes on their tails, and stem cells throwing tantrums when we mess with the system. But how does this all connect back to those ancient viruses we were talking about earlier, those ghosts in our genetic library?
That, my friend, is the million-dollar question. And the answer may surprise you, but we'll get to that right after the break.
And we're back. We're exploring the hidden world of ancient viruses embedded in our very own DNA. Before the break, we were talking about these tiny histone proteins and their mysterious modification sites, K9 and K27. And apparently messing with them really throws a wrench in the whole cellular machinery.
Yeah, it's like swapping a few lines of code in a complex program. Seemingly small changes can have massive, unpredictable consequences. And this is where those ancient viral remnants, those ghosts in the machine, come back into the picture.
Okay, you've got my attention. How do ancient viruses fit into all of this?
So scattered throughout your DNA and every human's DNA are these sequences called endogenous retroviruses, or ERVs for short. Basically, they're bits of viral DNA that managed to weasel their way into our ancestors' genomes millions of years ago.
Hold on. We're all carrying around bits of ancient viruses in our DNA. That sounds kind of freaky. Is that dangerous?
It's actually not as scary as it sounds. Most of these ERVs are harmless relics. Basically, they've been disarmed over countless generations of evolution. Think of them like faded photographs in a dusty old album, reminders of the past, but they don't really affect us in the present.
Okay, so these ancient viral invaders are mostly harmless souvenirs. But you said most. Are there some that aren't so dormant?
Yeah, that's the really fascinating part. It turns out evolution is even more clever than we thought. Some ERVs have actually been co-opted by our bodies to perform helpful functions, like regulating our own genes.
Whoa. So evolution took these potential threats and turned them into allies. That's amazing. So what does this have to do with our histone sticky notes and those mysterious canine sites?
Remember how we talked about those canine modifications acting like do not highlight flags on certain DNA sections?
Uh-huh.
Well, it turns out those flags are really important for keeping some of those ERVs in check.
You're telling me those canine sites are specifically controlling those ancient viral genes?
You got it. This new research found that messing with the canine site in stem cells woke up some of those dormant ERV sequences that act as enhancers.
Okay, time for a quick knowledge check. Remind me what enhancers do again.
Right, so think of enhancers like volume knobs for genes.
Okay.
They don't directly turn genes on or off, but they can dial up or down the activity of nearby genes. And what's really intriguing is that this research suggests the canine modification site is crucial for keeping those ERV enhancers turned down low, preventing them from disrupting the finely tuned symphony of gene expression.
So it's like our cells have this sophisticated security system to keep those ancient viral remnants from causing trouble.
Exactly.
And those canine sticky notes are a vital part of that system.
It all comes back to that library analogy.
All right.
Imagine if every book in the library was shouting its contents at the same time.
Oh my gosh.
It would be chaos.
Total chaos.
That's what happens when these carefully controlled epigenetic mechanisms get disrupted. Genes start firing off when they shouldn't, and that can lead to all sorts of problems.
This is incredible. We've got these ancient viral genes lurking in our DNA, and our cells have evolved this intricate system of histone modifications and enhancers to keep them under wraps. But why should we care? What's the big takeaway for us?
The implications of this research are massive, and they go far beyond just ancient viruses and stem cells. This is about understanding the fundamental language of our cells, how genes get turned on and off, how our bodies develop and function, and what happens when things go wrong.
Okay, so this isn't just about some abstract scientific discovery. This has real-world implications for our health.
Absolutely. Think about diseases like cancer, where cells start dividing uncontrollably, or neurological disorders, which are often linked to problems with gene expression in the brain. This research could unlock new ways to diagnose, treat, and even prevent these conditions by understanding and potentially manipulating these epigenetic control mechanisms.
That's huge. And I'm guessing this also ties into our immune systems, especially since those immune-related genes went haywire in the study when they messed with the canine sites.
You're spot on. Remember how we were talking about ERV sometimes playing a beneficial role in our own gene regulation? Well, the immune system is a prime example of that. Some of our immune defenses actually rely on those ancient viral sequences. It's like we struck a deal with these ancient invaders. You can hang out in our DNA, but you have to help us fight off other threats.
Wow, talk about turning the tables. So we've gone from dusty library books to cellular security systems to ancient viruses lending us a helping hand. This is seriously blowing my mind. But before we spiral down the rabbit hole any further, let's take a quick break. When we come back, we'll explore what this all means for you and the future of medicine. Stay tuned. And we're back. So we've been on this wild ride, right? From dusty old books to cellular security systems to ancient viruses lending us a helping hand.
It's pretty amazing stuff.
It really is. I'm still trying to wrap my head around it all, but let's bring it all home. What does all of this incredible science actually mean for you, the listener?
Well, that's the million-dollar question, right? Because at the end of the day, what we're talking about is the potential to revolutionize medicine as we know it. I mean, imagine a world where we can develop therapies that precisely target these epigenetic mechanisms.
You mean like those histone sticky notes and ERV enhancers?
Exactly. We could potentially switch off the genes driving diseases like cancer, or turn on genes that have been silenced in neurological disorders. It's truly groundbreaking stuff.
So instead of just managing symptoms, we could actually address the root causes of these diseases at the genetic level.
Exactly.
That's incredible.
Yeah.
It's like instead of just patching up potholes, we could actually rebuild the road itself.
That's a great analogy. And what's even more exciting is the potential to tap into the beneficial aspects of our ancient viral hitchhikers, too.
Oh, right.
We're already seeing how some ERVs play a role in immunity, for example. But imagine harnessing their power to develop new vaccines or antiviral therapies.
Wow. So you're saying that by understanding this ancient viral history and these intricate gene silencers, we could unlock a whole new world of possibilities for improving human health.
Absolutely. It's a powerful reminder that our genome is not just a static blueprint, but a dynamic library that's constantly being shaped and rewritten by evolution. And as we delve deeper into its secrets, we unlock some incredible possibilities.
This has been one mind-blowing deep dive. We learned that our DNA is a fascinating library full of secrets. Our cells have their own team of meticulous librarians, and some of our oldest enemies might actually be powerful allies in disguise. It's enough to make you look at your own reflection and wonder, what other secrets are you hiding? Until next time, keep exploring, keep questioning, and keep those ancient viruses in check.